Hi, good morning, everyone. I'm Diana, a PhD student from University Putra Malaysia. My research is all about exosome, looking at how exosome can be a better alternative to current diagnostic procedure for prostate cancer patients. The topic of my presentation today is optimization of exosome isolation technique from prostate cancer cell lines using ExoQuick-TC and protein analysis by Jess blot. There has been growing interest in exosome research worldwide. Exosome is applied in many areas in medical field, partly to be alternative to current diagnostic protocol, treatment in diseases, and disease monitoring purposes. Therefore, how exosome is brought from its original biological nest to bench is crucially important because it impacts on the isolated exosome quality and efficacy of the downstream applications. Here, I share my experience in isolating exosomes derived from prostate cancer cell lines via precipitation method using ExoQuick-TC reagent and how we optimize the isolation method. What is the significance of my study? Exosome can be a better alternative to prostate serum antigen or PSA and intrusive prostate biopsy in diagnosing prostate cancer. Exosomes excreted by primary and metastasized prostate cancer cells can be extracted from the blood and urine to determine the current clinical condition of suspected prostate cancer patient. Mimicking the biological situation, we cultured primary 22RB1 and metastasized prostate cancer cell lines such as LANCAP, PC3, and DU145. We then isolated the exosomes from the collected conditioned medium using ExoQuick reagent. There has been various exosome isolation kits in the market nowadays. ExoQuick hasn't been spared from little flaws, and we did optimization from there on. Below are my experimental works that I want to share in this presentation. After we isolated the exosomes from the conditioned media using ExoQuick reagent, we did exosome characterization. The exosomes were visualized on the transmission electron microscope or TEM for their morphology and size. Their size distribution was measured by dynamic light scattering or DLS system using Zetasizer nanoparticle instrument. The exosome surface biomarkers CD9 and TSG101, as well as endoplasmic reticular markers GRP94, were detected using automated Jess blot analysis. And finally, we did some optimization to the isolation method, and I'll show you the before and after results. Following the ExoQuick user manual, the collected conditioned media was centrifuged at 3000 gravitational force, or G, for 15 minutes at 4 degrees Celsius. The supernatant was then transferred to another new centrifuge tube, after which it was filtered through a 0.22 micrometer PVDF filter. Then ExoQuick reagent was added according to its ratio to the amount of conditioned media and left at upright position in a chiller overnight. The next morning, it was centrifuged at 1500 G for 30 minutes at 4 C, and then the supernatant was aspirated. The residual was centrifuged again at 1500 G for 5 minutes at 4 C. Finally, the fluid trace was removed and the exosome pellet was resuspended with 100 microliter of cool PBS. This is how exosomes derived from prostate cancer cell lines look like under TEM. They appear either single or gathered cup-shaped, rounded to oval frame particles. Some appeared shrunken and collapsed, with a mouth-like figure, having a slit or fissure in the middle of the vesicle. The end-to-end measurement of the particles ranges from 70 to 200 nanometer, which is within the predicted size of exosomes. The average size distribution of prostate cancer cell lines derived exosomes was measured using DLS systems by Zetasizer instrument. Size distribution demonstrates the percentage of particles of a certain size. Shifting of peak size distribution to the right indicates bigger particle size. In this case, PC3 has got the biggest average size distribution compared to other exosomes derived from other cell lines. There's possibility of particles aggregation because when we compared with the individual size appeared in TEM. The detection of common exosome protein biomarkers expression CD9 NTSG101 was performed by automated Jess Western blot. CD9 NTSG101 were only detected in exosomes derived from 22RB1 and LAN-cap cells, but not in PC3 and U145. To ascertain if isolation technique using ExoQuick reagent is comparatively efficient to the more established method, exosome purity was assessed by the absence of endoplasmic reticulum ER protein marker, GRP94. GRP94 protein was detected in exosomes from all cell lines but not the cells. Indicates that the precipitation technique using ExoQuick reagent produces clean exosomes. During Western blotting, the completely automated Jess capillary system performed the separation electrophoresis and immunodetection procedures following plate loading. We noted that the separation electrophoresis of the ExoQuick lysates were lagging behind the samples isolated by other established technique. This suggested that the lysate is more viscous than the other samples. According to the manufacturer's instruction, the mixture is centrifuged at lowest speed, 1500 G, for 30 minutes post-overnight incubation, then followed by another brief centrifuge at 1500 G for 5 minutes as the final step. We then did some modification to the original protocol, where we washed or purified the exosome pellet with 5 ml of cool PBS before the final centrifuge at 1500 G for 30 minutes at 4 degrees Celsius. Because of the polymer, which constitute the ExoQuick reagent, exosomes were thought to be stacking next to and on top of each other, as can be seen in the before washing picture. This chemically inert polymer appears in TEM as negatively stained homogeneous sort of a net, holding the exosomes together. In comparison to after wash, exosomes are seen more dispersed, scattered, and liberated, with no image of the net anymore. The polymer chunks that hold the exosomes within leave the DLS with false measurement that shifted the peak of 22RB1 size distribution to the right, which means bigger size. Post-purification, the 22RB1 size distribution peak had shifted back to the left, indicating exosome release from the polymer captivity, though in this case, exosome from LAN-cap cells did not show any difference. The excess polymer causing the lysate to become viscous owing to bogus protein complexes resulting in the separation electrophoresis of the lysates were lagging behind. Post purification with PBS, the time taken for protein separation electrophoresis was also reduced, and the lysate in the capillary moved downward at the same pace of the other samples, eventually generate less and hindered previous background, leaving the targeted signal peak distinguished. Compared to before wash, there were outstanding background or jagged peak, together with the targeted peak signal for CD9 anti-SG101, indicating detection of possible contaminating protein expression. In the final step of exosome isolation using ExoQuick, low-speed centrifugation was needed to recover a bag full of exosomes, which simultaneously clearing away the polymer in the discarded supernatant. Then the exosomes precipitate was resuspended with appropriate amount of PBS for further downstream experiment. This resuspension might be insufficient to remove all remaining polymer sticking to the exosomes. As this polymer is soluble and easily dissolved in water, so it could be washed and cleansed with PBS and spinned down again, even though we might be losing some of the original pellet. In conclusion, we could see the significance and importance of the optimizing exosome isolation technique, not only with ExoQuick reagents, I believe, because it affects the quality of the subsequent tests and experimental work, which might affect the effectiveness in diagnosing a disease as well. In my case, post-isolation purification with PBS gives notable differences between before and after purification. I thank all of you for listening. Thank you.